Hey scientists, I wanted to give you guys a quick lecture on the structure of DNA. So it's been a while since we've talked about DNA, but you guys can pause the video here if you want to take a look at the objectives of this lecture. I'm going to dive right in and you guys feel free to take notes in the document provided. But we know that DNA is a nucleic acid, just like RNA, and it's made up of these building blocks called nucleotides. So a nucleotide is this monomer that makes up this macromolecule, and it's made up of 3 different subunits. So here we have a nitrogenous base, in this case adenine, and is bonded to this 5-carbon sugar, which could be deoxyribose if we're talking about DNA specifically. And then it's bonded to this phosphate group. So collectively, all 3 of these different parts are considered a nucleotide. For the different nitrogenous bases that we see in DNA, we can see there are 4 different types, and we like to use our capital first letter abbreviations for each. So we have adenine, guanine, cytosine, and thymine. And these are the 4 bases that we'll see in DNA molecules. So in a DNA molecule, we see that these nucleotides, remember one of these monomers, are covalently bonded to each other to form these different strands. And this is just a small segment of one strand of the DNA molecule. And we can see these nucleotides are bonded together in a string. So some other observations that were made by scientists, this first one, Chargaff made an observation that the amount of adenine and thymine that were found in DNA molecules were always equal, and that wasn't dependent on the organism. He saw that the amount of A's and T's in human DNA were always equal, in fruit flies, in any other organism. They're always going to be equal, and we know that because we understand that adenine and thymine base pair together, they match. He also noticed that the other nitrogenous bases, cytosine and guanine, also occurred in equal amounts. So we call this Chargaff's rule, and that correlates with our understanding of how these guys base pair together. Another important scientist who contributed to our understanding of DNA is Rosalind Franklin. She took this very famous photo. It's called Photo 51. And she basically used diffraction to take this photo. And ultimately, with the pattern we see here, we can understand that DNA is a double helix. So from just this photo and this pattern, we understand that the nitrogenous bases are inside the double helix and the sugar phosphates are on the outside. And this photo was actually used by some other scientists, Watson and Crick, who were independently working to ultimately unpack the same question as what's the structure of DNA. And they were really struggling until they had access to Rosalind's photo. So Rosalind and Franklin, Watson and Crick were building this model, and they incorporated Rosalind's information into their model and ultimately won a Nobel Prize for their work. So here's a model of DNA. You guys have probably seen something like this before. Another feature of DNA is that it has anti-parallel strands. So if you look at these 2 strands in this double helix, we see one going down here and the other one going down like this, and the other one going in the opposite direction. And that just has to do with the way that these strands bond together, and this anti-parallel or opposite direction is how they best match up. So we call that anti-parallel. So when we're talking about what's happening inside the double helix, we know that those nitrogenous bases are stuck together and they're base paired together. So they're base paired through these hydrogen bonds. And we see that there are 3 different hydrogen bonds shown by these dotted lines that hold together the cytosine and the guanine bases. And then we have 2 hydrogen bonds that hold the adenines and thymines together. So these hydrogen bonds are actually disruptible, so we can peel apart the strand of DNA and work with one of the strands if we need to undergo DNA replication or something else using that DNA sequence.